Good afternoon. I welcome you all to the afternoon session, and it is a pleasure to have with us Dr. Manpreet Kaur. Ma'am is an assistant professor, Division of Agronomy, SKUAST, Jammu. And the topic chosen by ma'am for deliberation is modern techniques of plant nutrition for sustainable agriculture production. Ma'am, you are audible, and I'll handle the presentation. You may start, ma'am.
Thank you. Thank you, Majid sir. Now I am proceeding with my presentation. I introduced with the topic modern techniques of plant nutrition for sustainable agricultural production. Let me introduce topic with you. As we all know that plant nutrition is to study
Just a minute,
functions.
just a minute, ma'am. Just a minute,
Yes,
ma'am.
yes,
Is the presentation visible to participants?
yes, sir.
It's not
Yes,
in
yes,
full view, sir.
yes, it's visible.
Please continue, ma'am. You have to focus on
Okay.
me. Voice would be from ma'am, and the presentation is shared by me. Screen is being shared by me. Got it? Please
Okay.
focus on my presentation. Please, ma'am,
Yes,
continue.
second slide. Yes. We are moving to second slide. Let me introduce you with the introduction of the topic. And the plant nutrition is to study functions and dynamics of nutrients in the plants. If we are talking about nutrition, it is a whole process. How the nutrients move, how they move in the soil, how they move in the plant, and how they increase the plant growth, and what is their metabolism. If we talk about the list of the plant nutrients, the current list has been given by some scientists. It's nearby 20, which includes primary nutrients, including carbon, oxygen, hydrogen, and NPKs, and then secondary nutrients, calcium, sulfur, magnesium, and the list of the micronutrients. And most of this, we all know that carbon, oxygen, hydrogen has been obtained from the air, and nitrogen, phosphorus, potassium from the soil, and calcium, sulfur, magnesium. These all other nutrients also obtained from the soil. Now, if we talk about the problems, why to introduce this topic? Now, this is a need of an hour. We are introducing this topic because after Green Revolution, what happened? We have introduced some varieties, some dwarf varieties, whose requirement for the nutrients is much more, and so we have to increase the doses of the nutrients. We have abundant, there's an abundant application of nitrogen, phosphorus, and potassium, where the nitrogen application is more because we have, our main goal is food security at that time, no? But slowly what it has been seen that our farmers have been using these nutrients as a blanket application. When we are using these nutrients as a blanket application, then slowly and slowly there emerges some problems, like the problem of wastage of the nutrients, some other problem like eutrophication, driving force is nitrogen and phosphorus, and the least use efficiencies of these nutrients. Like if we talk about the efficiency of nitrogen and potassium, it is 30 to 35%. Whereas phosphorus is 15 to 20%, and the secondary nutrients is 8 to 10%, and micronutrients have the minimum efficiency of 2 to 5%. So we have to think about that, what to do so that these problems should be addressed. Moving to the third slide, third presentation, if we see this table, what is the third presentation? I think you are moving the presentations.
There is no movement. There is no movement. It's in first slide only.
Yes, yes. Majid sir, are you moving the presentation? We are on the third slide. Okay. Let me move to the third slide. But my third slide is about the available form and mobility of these nutrients and their role.
Ma'am, tell me whenever you want to move to next slide. Let me know when to move.
Yes, yes, sir. We are in the third slide. Please move to the second slide. Na? I'm telling you, second slide. You are in the first slide. You are in the then third slide. Move to the third slide, sir, please. Sir, move to the third slide. Okay, if we talk about these slides, we have seen that the available form of these nutrients, their mobility. If we talk about the mobility of the NPK, they are very mobile. If they are very mobile, that their deficiencies has been yes, their deficiency. Please somebody contact Majid sir and ask him to move the slides if someone can do this.
Ma'am, tell me when to move to next. Don't just move. Keep on telling me when to move next.
Majid sir, move कीजिए please. Please move the slide. We are on the third slide. Please move to the third slide.
Is it visible now? Just a second.
And yes, this one. Okay, if you see this table, you will see that these nitrogen, phosphorus, and potassium, they are highly mobile. If they are highly mobile in the plant, then their deficiencies has been seen in the lower leaves. Whereas, if we talk about the micronutrients, they are immobile. Next slide, sir. Next slide. If we talk about the micronutrients, then they are immobile. If they are immobile, then what happened? If they are immobile, then what happened? Their deficiencies has been seen in the top. This is the reason why the deficiencies have been noticed in the top and in the lower leaves due to the mobility potential of these nutrients. If we talk about the role of these plant nutrition, mostly the primary nutrient nitrogen helpful in carbohydrate and protein synthesis, phosphorus in root growth, and potassium in immunity. If we talk about calcium, sulfur, and magnesium secondary nutrients, they are mostly the integral part of amino acids and seed production, as in sulfur, in calcium in cell division, and magnesium is a part of photosynthesis. If we talk about micronutrients, they are mostly involved as an enzymatic and catalytic reactions of photosynthesis and respiration. Now, I have already talked to you about why we have to introduce these modern techniques to control the nutrient exhaust and to reduce the emission of greenhouse gases and to reduce the problem of eutrophication. So, these modern techniques are very helpful to mitigate the problems of climate change. We take in the nutshell to meet the proper requirement of the crop as required by the crop, not by the soil. And in this modern techniques, we have feed to the plants, not to the soil. This is the main principle. Next slide, please. Next slide. Yes. Now, if we talk about the modern techniques of plant nutrition, first technique is nutrition on the basis of soil test basis. Now, I have already told that previously the application has been made blanket. Blanket means we are for the RDS. Whatever the package and practices suggested, we are going for the blanket application. But no to this. We have to apply now we have to go for nutrition on the basis of soil test. This is an experiment which has been conducted by one of my student, MSc student, and what we have in this experiment, we have worked on different varieties and the INM, Integrated Nutrient Management. Here we have provided our nutrients on the basis of soil test. First, we have taken the composite soil samples and then we analyze the nitrogen, phosphorus, potassium and put our initial values. The initial values have been given which suggests that the nitrogen is at the low, phosphorus and potassium are medium. So, it means from the initial value, it strikes in our mind that yes, we have to go for more nitrogen. But how much nitrogen? It has been calculated with the help of the model named as fertilizer calculator. We have put our values, initial values in the fertilizer calculator and then it suggested how much nitrogen and nitrogen is added more. This table is clear that different varieties have been taken and the Palampur one and JHO 851. They are the promising varieties. These are the three promising varieties of the oat fodder, and their requirement is same, and their Palampur one and Kent requirement is same, lagbhag, and JHO 851 has a higher requirement. So we have maintained the fertilizer requirement according to the need of the crop. According to the need of the crop, we have then increased the RDF values as per the values calculated by the fertilizer calculator. Now, moving to the next slide. Next slide, sir. Move to the next slide. Yes. This slide, I think this is my personal opinion that this is very important. This diversification is very important technique in modern plant nutrient. Why so? Because the choice of the crop is to be done very judiciously, because our Indo-Gangetic plains where we are living is most have this predominant cropping system of rice and wheat, and it is putting lot of pressure in our resources, in fertilizer, etc. Now, this crop diversification has been an important source to mitigate these problems of losses in fertilizer and also to restore the crops, restore the soil with the restorative crops, because this along with rice wheat is an exhaustive system, it is the prevailing system. No, we actually from 2006 to 2010, I have worked as a technical assistant in AICRP IFS. At that time, where we are conducting 5 experiments, and one of the experiment is this diversification. It is given here. Then another is integrated nutrient management. It is in our next slide, and another experiment about organic that is also covered in this lecture, and 2 more experiments. Then what we have, the results are, these are the long-term experiments having a duration of 8 years, 20 years, 15 years. So these results are very much beneficial. So I have put these results in my slides, because this diversification we have seen that if we substitute wheat with other cropping systems, because we can't ignore rice here. Rice is the principal crop here, a staple food. So we have substitute wheat here. And when we substitute wheat with another cropping system, we have seen the beneficial results. Next slide, please. And we have seen that in this cropping system, rice, marigold, French bean, it is the most economic system in our subtropical zones, where with a great system profitability, having a great production efficiency, moreover the apparent nutrient productivity means how much kg per hectare yield productivity is achieved by a unit kg of nitrogen. That is also very good results comparison to rice with the prevailing system. So we have recommended this technology about the rice, marigold, French bean. Next slide, please. If we see the role of diversification in the soil study, we have also seen that these incorporation of Berseem and green gram like crops, leguminous crop, have also built up the organic carbon, maintained the nitrogen, phosphorus and potassium over the initial status. You can compare this initial status from initial status, the results are becoming good. There is a definite build-up. Okay, this next slide, please. Now, another technique which we have to use is INM, Integrated Nutrient Management. This is another technique, because we should not depend on only one source, that are the inorganic fertilizer, nitrogen, phosphorus, potassium. We have to go for integration. Okay, when we are going for integration, we have to use all the possible sources, all the organic, inorganic and biological components of nutrient sources as in an integrated manner. This is a slide of same from ACRIP IFS. This is a long-term permanent experiment. Here the T1, 12 treatments are there. One is T1. T1 is just control, and T12 is farmer practice. And another is the different gradient levels of fertilizers used. And the T6, here the T6 is profitable. Here the T6 is purely a INM technique, where we have 50% recommended NPK through fertilizer and 50% NPK through farmyard manure. Wherein the wheat The 100% recommended NPK through fertilizers. Main interventions has been done in rice. Next slide, please. It has been seen this T6 treatment has a better results in the form of productivity, system cost cultivation, benefit cost ratio, and moreover, next slide, please. This is a very important graph. The readings have been taken. Yes, next slide, please. The readings has been taken from 1985 to 2012 by pooling 3 years data. You see, in the initial stages, what happened? All the treatments are very close at a particular time. They're very close, but slowly, when we reach further from the 1985, from 2012, if you see 2012, T6 is increasing in soil organic carbon. T8 is also good, and this control is least, and then we say this farmer practice is least. So if we go for integrated nutrient management, the results are slow because the release of these urea NPK in the form of inorganic is very quick. They quickly release the things, and so in the first, second year, the results are good in these in these treatments where we have used purely inorganic fertilizers. But slowly and slowly, when we move far away, then what we see, those treatments are excelling where we use organic inputs. So this is the finding. Next slide, please. Okay, this is another trial. This is a trial in the temperate conditions, because our Jammu and Kashmir is gifted with 3 different types of zones. One is subtropical zone, another is temperate zone, and one is intermediate. I have also been posted from 2010 to 2014. I have been transferred to subalpine zones of JLK, that is the Bhadrawa. In that that period, I have conducted this trial of INM in that conditions of subtropical, temperate zones. And what we have seen, because the farmers of Bhadrawa, the hill tracts are default organic, and the farmers of the Bhadrawa are mostly practicing rajmash and maize intercropping system, and they are using the organic sources, and they just spray urea. There is no particular technique of rajmash and maize intercropping, as well as they are not using the proper doses of nutrient management. As maybe you know that the rajmash of Bhadrawa is very much popular. They have a good export value and they are so tasty. So the great potential of J&K's where the saffron is very famous, the rajmash of Bhadrawa, they are also very famous. So we have to work mostly on the rajmash crop and how to excel its yield potential. So there is a thrust to first to give a proper cropping system, and here we have given this maize plus rajmash 1:1 cropping system excelled among all the systems, and the farmers of those regions are just they are not maintaining any rows, and they are just going for mixed planting. They mix the maize and rajmash seed and they throw it in their soil life. But we have recommended them to go for line sowing, and the result is that we get the more benefit cost ratio there. And in fertility management, the results are promising where we have used 75% RDS with 25% night farmyard manure. Next slide, please. The same results are validated because these are the good results. We find a new technology here and we want to validate our results. So we have also conducted OFTs by existing recommendations which are present in our package and practices, where there is a mixed cropping system given in your slides 8:1, and with the inorganic sources of fertilizers, and with the new recommendations where we have to use 1 row of maize with 1 row of rajmash and with the INM treatments. And what we found that where we have used, next slide please, where we have used our new recommendations, the results are good, the BC ratio is good, then the existing recommendations. So on the basis of this, we have changed our previous package and practices and use this new recommendation. Now our package and practice is containing these new recommendations where we have used this in INM, farmyard manure plus inorganic sources 75% plus 25%, and where we have also used the cropping system, 1 row of maize followed by 1 row of rajma. Next slide, please. Now, next slide is about the modern nutrition by organic sources. So, this is another alternate source of conventional inorganic, and this is a trial which is conducted in ECRIP IFS, and here we have taken the 8 treatments, and what we have do from T2 treatments, we have given 100% organic sources to our field, where we have used the sources as farmyard manure, vermicompost, and non-edible oil cake. And then T3, T4, and T5, T3 and T4 are just addition on T2, where we have to manage our pest control, weed control, and trap crops are used. And T8 is a dummy plot where we have to replicate the best treatment. So T2 is followed in the T8 plot. Next slide, please. And what we have seen in the results, T2 is where we have used organic sources has excelled. They have excelled after 2-3 years. They have proved to be excelled the recommendation. And similarly, next slide, please. There is also found a change in soil properties and soil microbial counts, and they have also increased with the time over the initial status. So we recommend that organic nutrients, they are good. No, next. From 2014 to 2019, I have again transferred to division of ILFC at Arispura campus, where I have to work as a fodder specialist, and because there are 50 dairy animals, where we have two mandates for me to I have to work on 2 objectives: one to manage feed and fodder for them, and another to recycle their waste. And the wastage is for the 50 animals is huge, and we have a challenge to produce each and every part of their dung into compost, and we have made a unit of vermicompost, and I have given, because vermicompost is the main portion of the organic source. So I have also given the slides how to prepare vermicompost. We have prepared vermicompost here. First, if somebody is interested to prepare a vermicompost, then he has to what to do. First, we have to select a shady place, because our worms, we have used Echinna foetida here, because Echinna foetida is the most promising species of earthworm in our subtropical zone, because its multiplicative rate is so high. And we have first of all choose a shady place. Why shady place? Because these our earthworms, they are very much phobic to light, and they don't respond good in light. So shady place is must. We have make arrangement. You see the ceilings of our shade. We can also use low technology techniques, but we have made here. What we say that this teen is made, so but we can also in another structure. This is a starting structure of our, so we have used this. But when we have get familiar with these conditions, then we have also made our compost as a heaps, but just spreading a rice straw on it, so to provide a shade in the low cost. We have also made that photographs have not included in here, I have forgotten, but that is also a technique to maintain a shade. Okay, so then the next step, shady place. Then we have to bring a dung which is 10 to 15 days old, because when the fresh cow dung, it has very much great heat, and there is a proper requirement of the heat for the earthworm. They don't survive in a more heat above 40 degree, 45 degrees. So the fresh cow dung has a great heat, and so we have to make it cool for at least 10 to 15 days. Then we have to put it in these trenches, which is the dimension of a trench is just 10 meter in the length, and they are 75 centimeter in the depth. And because why 75 centimeter in the depth? Because the earthworms only move up to the 75 centimeter or 1 meter. They don't move more than it. So the depth is kept low here. Then we have to repeat the layers of cow dung and because when we have to go for composting, then we have to see that C/N ratio should be maintained. C is carbon and N is nitrogen. For nitrogen, green is used, and for carbon, straw, some straw and some whatever say the dry substances like straw, like newspaper, like cardboard. But we have lot of straw. This cow dung also contained the bedding material for our animals, so it has enough of the straw mixed with it, so we don't use more of it. But for making vermicompost, there is one layer, 15 centimeter layer of cow dung, then followed by 15 centimeter layer of some carbon source like straw, mostly straw, or mostly dry leaves of the trees, or mostly weeds, dry weeds, whatever brown, it is used as a 15 centimeter. Then we have to follow this layer. Then we put 1 to 2 kg of earthworm in this long trench, and surprisingly, after some time, we found it's a great population of this earthworms. You see in the fifth slide, our laborer is holding the ready compost with lot of worms. One thing is very important. The third picture, we are using trungles to turn the compost. This is very important. Turning is very important, and because more turning, more oxygen, more oxidation, and more rapid the process is. And the last is sieving. Sieving is done. These are the boys. We are also giving training to them, and this is the picture of those trainers who are taking trainings at that time. So this is how to prepare vermicompost. Next slide, please. Yes, another slide is talking about the treatments which are taken under the temperate condition. When I am there, I have finished that one INF tree trials, and I have proposed another trial about the organic treatments. Because in the subalpine condition at Bhadarwah, there is the two main things which get waste more. Pine needles. Pine needles, because it is subalpine zone and the pines are available in the plenty there. And we have to make something about the pines, because pine needles, they have a good source of NPK. And moreover, pine needles have to be composed. So there the new techniques we have made is phosphor composting. We try to compost the pine needles as well as rice straw. Now, the T6 treatment here is given. Because T1 is RDF, T2 is half RDF, half farmyard manure, and T3 is about T2 plus mulch at pine straw, mulching of pine straw, because we try to use the wastage of pine straw, because the pine is the wastage there. We want it as a source of nutrients. And next we are using vermicompost and phosphor composting through rice straw, and then again vermicompost and phosphor composting through pine straw. Then in T6 we have made the recommended RDF by farmyard manure, vermicompost, neem cake, and pine needle. And in the T7 along with T6, where all the organic sources, farmyard manure, vermicompost, neem cake, and pine needles have been used along with biofertilizer, rock phosphate, and phosphorus soluble microorganisms. So this is the organic treatment. T8 is where the trichoderma is used, and cow urine, because we have to control our pests there, diseases there. For that, in the one treatment T8, we are using cow urine, 5% foliar spray, a 15-day interval. And in the T9 treatment, we have used neem oil, 3% foliar spray. And then I get transferred, and this project ultimately goes to Dr. Rohit Sharma, and he has given the results. T7 excelled in all the treatments with higher grain yield and rajma yield. T7 is there where we have used organic sources plus biofertilizers plus rock phosphate and plus solubilized microorganisms, and its properties, soil properties are also found good in those treatments. Now, this is the methodology. Yes. Next slide, please. Next slide, please, sir. Again, next slide. Next slide. Phosphor composting. Yes, sir. Move to next slide. Yes, this. This is the main thing there, because we have a lot of pine straw. This is a problem there, and it gets waste. We don't want to be waste the pine needle, and we have started making composting of pine needles. You see, it's very easy to make phosphor composting. These pine needles, they are collected and they are then put in. We have used indigenous sources which are easily available now in this huge, what we say, bucket, and like now, and it's a made up of this. In this bucket, we have put mud slurry, about 1 kg cow dung in 1,000 liters of water. This is the recommend. This we have given, and in this we have dipped our pine needles for 10-15 minutes. Then take it out. Then we have make a pit of proper dimension, depending upon the material we have. Then we have make an aeration by putting sticks in it. You see the 5th slide where we have put using sticks. Then we put layer wise this pine needle, and then sprinkle rock phosphate. Rock phosphate, we have brought this rock phosphate from Jammu, and this rock phosphate has a 6%. We use this rock phosphate 6% of dry matter basis of pine needle, and also for the rice straw. And then we have covered this our pit with polythene. Why polythene? So that the moisture get maintained. After 90, say 120 days, we this start becoming 120. Almost it takes 120 days, because under temperate conditions, the temperature is different, and the scenarios are different than tropical, and so we get this in 3-4 months. Okay, next slide, please. Yes, biofertilizers. We have also used biofertilizer in this organic treatment. Biofertilizer is also a compost, phosphocompost, vermicompost, biofertilizer. They are the important inputs in organic agriculture. In organic nutrition, this is the important components. So another important input is biofertilizer. So biofertilizer can be nitrogen fixer, they can be phosphate solubilizers, and they may be symbiotic, they can be free living. Next slide, please. And we have seen, if we talk about nitrogen fixers, then it may be rhizobium and azotobacter for non. Next slide, please. Okay, if we see this slide, and we are preparing in the Badarwai, we are preparing this biofertilizers and inoculating biofertilizers with rajmash. In 1 beaker rajmash is taken, another beaker this maize seed is taken, and we are putting biofertilizer in both the beakers, and then we have made the jaggery, and we are putting jaggery in the next slide, and then we stir it, and then when they get mixed properly, then we have to shade it. We have to keep it to dry under the shade. We have kept it under the shade so that they get dry, and then we use these seeds. Now, third slide is the use of trichoderma. We have also used trichoderma in this organic trial. Next slide, please. Now, another technique of nutrition, modern technique of nutrition is use of liquid fertilizer. If we talk of liquid fertilizer, then what we see, there is a trend of vertical farming, because more and more we have a shrinking holdings, and our day by day our holdings are getting fragmented. Moreover, our farmers are mostly small and marginal farmers. About 86% of our farmers are marginal and small. They have a little holding and also there is a more pressure on urban. The peoples are migrating from rural areas to urban, so there is more pressure on urban areas, and if we want to grow vegetables and crops there, then there is no other choice than the vertical farming. So vertical farming is suggested in those areas where there is a landless laborers, and we have to suggest these techniques on those areas. The three techniques are mostly used in this vertical farming: hydroponics, aeroponics, and aquaponics. If we talk about hydroponic, in both in these three types of the vertical farming, in all these three types of vertical farming, what is we are using? We are using the liquid source of fertilizers. If we see the picture of hydroponic, here given the picture of hydroponic, these are the soilless media. We use the media like perlite, coconut coils are used here. We can use vermiculite here, having rich in potassium, and these are the media. These medias are used. We can't use soil in these media. We use these types of media in these type of system, in this type of vertical farming system. If we see that there is a tank and it will provide drop by drop water to the pots provided in the vertical stack layers. This is the principle behind the hydroponic. Please move to next slide. Now, if we talk about the aeroponic system, then in aeroponic, what is done there? The roots are suspended in air and they are provided with the mist nozzles, and this is called aeroponic. Mostly the people prefer hydroponic. Aeroponic is sensitive; some disease may occur, and great care is to be taken when we are going for aeroponics. So hydroponic is more famous than aeroponic. Next, please. If we talk about the aquaponic, this is the aquaponic. This is the third type of the vertical farming system. This is the third component of vertical farming system. Here, what happened? Fishes are used, and these fishes secrete their fecal matter, and their fecal matter get recycled, and it is given to the vegetables. So this is the filtered nutrient which our vegetables used. So if we say about the popularity, I can say that hydroponics are getting more famous because they're easy to handle. Hydroponic. If we talk about aeroponics, some results have been shown that the disease-free tubers of potato can be grown as an aeroponic, because our potato has a problem of infection because tubers are required which are disease-free, and aeroponics are mostly used in those tuber-free potato, growing tuber-free potato, whereas hydroponics are mostly used in many vegetables. Now, like there is a trend of producing hydroponic fodder. Next slide, please. No, the crops suitable for vertical farming are mostly the green leafy vegetables. They are supported by these vertical farmers, and some medium vegetable like cabbage, cauliflower, tomato, brinjal, and then big vertical crops if we talk about, and they are mostly we have put our hand in fodder crops, maize, sorghum. The hydroponic fodder is now introduced, and it is, I think all of us are introduced by this hydroponic fodders, and it has also very great nutrients value. This is a structure, this is a picture. It is about a picture of SKUAST Jammu. Here low cost hydroponic unit has been set by one of the doctor, Dr. Gurdev. So he is working on this hydroponic, and he has also found some promising results here for the landless laborers. Next, please. If we talk about what happened, it has been given by the scientists. Some less work has been noticed in this vertical farming, but whatever the work there is, it has been shown that these are better in chlorophyll content, carbohydrate content, and protein content than the conventional method of the sowing. So we say that the hydroponics, they are preferable than the another system. It means vertical farming. If we go for vertical farming, there may be a better nutritional value of this vertical farming as compared to conventional farming, because there is less wastage of water, less wastage of nutrients, uptake is great because it is under controlled condition. Mostly these vertical farmings, they are made under controlled conditions. When something is in controlled condition, wastage is less, assimilates more, uptake more, source sink relationship more, so it is better in quality than the conventional method. but it is costly. This is the con of this. Next slide, please. Another technique, another high-tech technique is site-specific nutrient management. Its main principle is to increase nutrient use efficiency. It is governed by 4R, means right product. It depends on soil testing, balanced fertilizer, enhanced fertilizer efficiency, right place. Matlab, we have to give our fertilizer at right place, at right time. The time should be right and at right rate. What to do? At the right rate. The different tools are used in site-specific nutrient management, because site-specific SSNM is coming under precision agriculture now. Na? So precision agriculture, where we have to precise the things. Now we have to given the things according to their need-based feeding of the crops is given in precision agriculture, in precision farming, and in site-specific nutrient management is a part of precision agriculture. So we have to give our crops a need-based feeding of crops. So different tools are also used in this SSNM, because GPS. One tool is GPS, GIS, RS, VRT, farmer laser land leveler, leaf color chart, green seeker, and chlorophyll meter. If we talk about the GPS, GPS is a global positioning system. What GPS main function is? It tells about the location of the thing. It is very important. It tells the location. Okay, when this GPS, the data collected by the GPS, data collected by the chlorophyll meter, data collected by the remote sensing is used in GIS. GIS is a broader term. It includes all these systems. It takes the data from all these systems, and then it gets image processing, then it gives the results. GIS is more informative. GIS is the geographical information system. It also provides data imaging. You know, what type of soil, what type of topography. It tells much. GIS is the main. And then the land leveler. We all know the land leveler. When we do laser leveling, it has a great benefit. The water gets saved. There is an increased source of nitrogen and water use efficiencies, and the cost of production gets reduced. Next slide, please. No, another SSN approach is use of. Next slide, please. Approach is use of leaf color chart. Next slide, please. Yes. Leaf color chart, we all know that we match the leaf strips with the different what we say plates that is given. And when in the back of this strip in the leaf color chart, there is a given sufficiency level or deficiency levels, and according to that we have to top dress our nitrogen. Next is about the use of the green seeker. This is more modified, and it has a plasma in it, and it gives us NDVI. NDVI means normalized difference vegetation index. It gives the NDVI of the biomass. When it gives the NDVI of biomass, then we are able to know that whether our plant is healthy or not. If our plant is poor in nitrogen, etc., then we apply nitrogen depending upon the NDVI values, whose value range between 0 to 1. If the value is greater than 0.5, it means efficiency, but there's no deficiency. If it is less than 0.5, then we think about to top dressing our fertilizer. Next SSNM tool is chlorophyll meter, and this chlorophyll meter, it tells about the chlorophyll present in the plant, and it gives its value in SPAD. If the SPAD value fell to between 29 and 32, it indicates that additional fertilizer was necessary. It's how we have to use these techniques. Now next technique is next technique nutrition by, this is the seventh technique. Yes, seventh technique, sir. Yeah, yes, this one. Nutrition by using technique of nanotechnology. Now, there's a trend of nano. If we see the mobile has been changed to nano, if we see the television have changed to nano, so it is a period of nano. And nanotechnology has also been given a great role in the nutrient management. Now we are using nanoparticles. Nanoparticles are those which have a range of 1 to 100 nanometer. And what is the principle behind it? When we go for the nanoparticles, what happened? They have more surface area over the volume, and they have more reactivity. So when they have more reactivity, there is the less losses. So in case of nano fertilizer, they are divided into slow nano fertilizers and specific release of nano fertilizers. And depending upon their classification, they are nanoscale fertilizers, nanoscale additive fertilizers, nanoscale coating fertilizers. Nanoscale fertilizers are made up of nanoparticles which contain nutrients. They are just nanoparticles. And nanoscale additive fertilizers are those traditional fertilizers which we have used nanoscale additives, and nanoscale coating fertilizers are the traditional fertilizer which are coated or loaded with nanoparticles. If we talk about the synthesis of nanoparticles, two approaches are used. One is top down, another is bottom up. Top down means we have to break the big particle into smaller to create a nano size, and another is bottom up, where we have to make a smaller particle to a big particle by recognizing their molecular recognition. Top down mostly involves the physical type of system. Like we say in the top down, we use ball milling, laser ablations. We have to break the thing. In bottom up, we have to synthesis the thing. We have to make the thing. So in bottom up, we use the chemical processes like sol gel method, solution to gel method, and then precipitation method. Like the chemical methods, we have to synthesize the thing. So chemical methods are used in bottom up approach. Next slide, please. So if we talk about the why nano fertilizer, then we see there are definitely advantages of nano fertilizer over conventional methods. Their staying power is more. Nano fertilizer can stay for more than 90 hours. If we talk about urea, it can stay 90 hours as compared to conventional, which can stay nearly by 25 hours. If we talk about phosphate fertilizer, it stays for 40 to 50 days as compared to conventional 10 to 12 days. Also, due to these more surface area over volume, it increase the uptake and assimilation and nutrient use efficiencies. It maintains the leaf dry weight, germination, assimilation increase, physiological and biochemical parameters increase, enzymatic activities increase, growth hormones synthesize more, and more quality produce are obtained. So this is due to easy penetration of nano fertilizers. What are the different tools used in nano fertilizer? They are nano biosensor and multi-walled carbon tubes. Nano biosensors are placed, and when they get the reaction in the soil that there is a deficient, then we have to supply the nano fertilizer. These are the role of the biosensors. Similarly, the role of multi-walled carbon tubes, they also act as a sensor in the soil. Next slide, please. This is the most important, this is the important slide because this is the soil test crop response based on target yield equation. This is STCR. STCR is an equation based nutrient application where first the target yield is recommended. The formula is given here. This is the fertilizer dose. We have to formulate the equation. We have to make the equation for different phosphorus, potassium, and nitrogen here. Formula is given here. You can see the formula. It is the nutrient requirement multiplied by target yield into 100 divided by CF. CF is the percent nutrient contribution from fertilizer, and then subtracted by CS. CS is the percentage contribution from the soil, and then divided by CF, and let's multiply by soil test value. Next slide, please. Next is how to calculate the fertilizer dose required for soil test phase. It has been given here that if the targeted yield is 1.5, fifth point, if the targeted yield is 1.5 ton per hectare, then we have to give fertilizer of phosphorus fertilizer is given here, how to calculate it, and then the fertilizer for phosphorus should have the value of 12. If for the 1.5 ton per hectare yield, we have to go for fertilizer requirement of phosphorus is 12 kg per hectare. It is given here. So how to calculate? This is an equation based. Next slide, please. Yes. This is another onion crop is taken here, and there are three equations I have given: fertilizer nitrogen, fertilizer phosphorus, and fertilizer potassium. Basic data is given, and we have to target yield is given. Whatever the target yield, we have to put the value of target yield here, and then we have SN means soil-based nitrogen, soil available nitrogen, Sp means soil available phosphorus and soil available potassium. So on the basis of these equations, we can find out how much amount of fertilizer we have to need it so that the targeted yield is obtained. So soil test based recommendation, जो crop recommendations होती है, they are mostly based on equations. To first we have to set the target yield. Next slide, please. Here is a clear example how to get, next slide is, how to get the 225 quintal per hectare target yield of onion. You have seen that different treatments have been taken here, but T3 treatments have a, if we see the last column where the percentage variation in the targeted and actual yield is given, you see that the less variations have been obtained in T3. It means here is we are successful to get 225 quintal per hectare of yield when we are applying fertilizer dose 96, 102 and 53, and the soil test values have also been given here. So this soil test value recommendations have been given. First we have to set our target yield, then we have to set the equations and we calibrate the equation, and then we have to go for validations. Next slide, please. This is the last technique. Anji, next slide, Hanges. Nutrition based on techniques of remote sensing, imaging technologies for plant nutrition. So, in this, it has been seen that what is the remote sensing? Remote sensing means without touching, we are getting the information about an object. What is the principle behind it? How we get without touching, we get the things familiar by the. It is a principle of reflectance. Everything has its own reflected energy. Na? For example, if we take the case of crop, canopy of the crop, we see that the nitrogen deficient or phosphorus deficient crop has a different reflection as compared to the rich crops. So when this pattern is used, so every crop made its own specific spectral signature. This word is very important, specific spectral signature. Every crop made its own specific spectral signature depending upon its reflected, absorbed and transmitted energy relationship. So this is used in remote sensing. This spectral signature is used on, and then the images are made with the help of GIS, and then I have already told you that GIS collect the information from different sources, from GPS, from remote sensing, and then it go for image processing, and then we got the thing whether we have to apply more nutrients or we have to, what we have to do. So we can conclude here that due to the problems, due to the changing scenario under climate change and more problems of food security, we have to shift from blanket application to those modern techniques of nutrition which can increase the nutrient use efficiency as well increase the productivity, apparent nutrient productivity of the crop, and also we have not to emit more greenhouse gases. We have to control greenhouse gases, and we are able to control greenhouse gases when we are stopping the wastage of nutrients. Mostly we know that carbon dioxide, nitrous oxide, they are the major culprit in the climate change scenario because they are producing they are produced from the wastage of the nutrients. So we have to stop this. We have to stop the blanket recommendation, and we have to switch to INM, organic nutrition. We have to go for precision farming, SSNM tools, STCR. We have to go for the SSNM techniques, nanotechnology. We have to integrate all these things. Biofertilizer should be used. Liquid fertilizers in hydro-aeroaponics can be used. So we can say that we have to think about the things in totality, and then we have to decide what to do. Thank you very much. Thank you, Majid sir.
Thank you, ma'am.
Yes.
Now, if there are any questions, ask. We have a few minutes, then we can consider the session to be concluded. Yeah, there is a question in chat.
Okay.
Can you see the chat, ma'am?
Chat. Yes, plantation, these modern techniques, it includes all the basic things also. Now, because when we have to, the question I think is that modern techniques are applicable to plantation crop production. Now, so if we talk about plantation means specific your plantation crops. If you're talking about tea, coffee, or if you're talking about, I want to discuss with like INM techniques, they are used to every crop, na. Organic techniques they can use to every crop, and if we talk about the nano fertilizers, some techniques are universal. If we talk about hydroponics, aeroponics, less work has been done in the aeroponics and hydroponics. Yes.
Yeah, continue, ma'am. Continue. Not.
Yes. If we this some INM, organic nutrition, and this SSNM, this remote sensing, these nano fertilizers, they are the universal techniques. They can be used by everyone except the vertical farming techniques, because some less crops less work has been done now, and now the researchers are doing work and making them exploit these techniques. But we have just get success on some green leafy vegetables and some tuber crops, and the results we are awaiting for some results. So I can't suggest this technique, hydroponic vertical farming for plantation. Some other techniques can be used easily. Yes.
Any other question?
Just give me half an hour.
There is someone who wants to say, ma'am, can you share your email address? Yeah, ma'am, can you share your email address with them?
Yes, sir. Okay.
For future queries.
Yes, sir. You can share my mail ID. You have my email ID too.
Okay.
Yes.
I have put the email address in chat only. Please check.
Okay.
Any other question? Anyways, ma'am, I think this is enough for today's session. It was such a lovely presentation. Thank you. And on behalf of the participants and on behalf of the organizing committee, I express our gratitude to you for being with us. Thank you.
Thank you, sir. Thank you, Manpreet sir, for your support always. Okay, thank you.
Okay, thank you. We can join back tomorrow at 11. Thank you.